Hi, everyone. So it is 12:15 EST, so happy afternoon or morning, depending on where you are or whatever time it is for wherever you are in the world. And what we're going to do today is talk a little bit about genetics. And I'm calling it 101 and 201 because we're going to do some basics and make sure that you have some of the vocabulary down that you're hearing, especially when we talk about clinical trials and strategies. And also give you a little bit of insight into some of the research that's going on with that information in here. So let me start the slides. And all right. Okay. Can I get a heads up from ASF or someone that you can see the slide?
We can see your slides perfectly, Doctor. Thank you so much.
Okay. So, I guess the only thing I can't see is the chat box, which we were talking about maybe needing to see. So
it's okay. I can help you. I'll copy the stuff. Any questions that come into the chat, I'll copy them over to the Q&A for you.
That would be great because I can't see the chat. So perfect. All right. So let's get started. So my name is Becky Burdine, and I am a professor of molecular biology at Princeton University. But the reason I'm sitting here in front of you is because my first child was born with Angelman Syndrome, and that is Sophie, which you can see here. This is her riding her bike for the ASF virtual walk this year. Sophie is 15 and she is deletion positive. She has the big class 1 deletion. And I'll talk to you a little bit more about Sophie as we go along. These are my current disclosures. So currently I am a board member at the ASF and I am on the ASF Scientific Advisory Committee. I have also been helping Ovid with their clinical trial steering committee for STARS, Neptune, and Alara trials that they were conducting. I have also been a member of FAST, and I was their scientific advisor from the—
Too many screens going on there, huh, Drew? Too many screens.
Say again?
Yeah, but depending on how she has her set up on her end, she might have things that were blocking it, and she might not have been able to see it.
So I can hear you guys talking. Do you need me to— is everything okay?
Everything is fine, Doctor.
Sorry. Okay. All right, so let's go forward. So what we're going to do today, we're going to talk about the genetics behind Angelman syndrome. We're going to talk just briefly about what that means for you if you're a beginner. So, where should you be focusing your time? Then I want to talk to you about why we think therapeutics are possible, better therapeutics. And then a little bit about clinical trials and some things that you should know in general as clinical trial participants. Okay, so first we got to start with some definitions. And we're going to do basically turn everybody into little molecular biologists for the moment. So the thing you have to keep in mind when we're talking about genetics is that DNA, deoxyribonucleic acid, or DNA, contains all of the instructions for life. So what do we mean by that? Okay, so as you know, cells make up every part of your body. There are the neurons, which we'll talk about a lot, but all the cells, your skin cells, your lung cells, your muscle cells, all cells have a basic architecture to them. And inside each cell is a compartment called the nucleus. The nucleus is important because this is where all of that DNA is kept. It contains the instructions, and it's kept in these large structures called chromosomes. So if you go and focus down in on a piece of a chromosome, you'll see DNA, and it's always represented in this helical structure. The little rungs of the ladder between the helix is the information that the DNA has, and the cell knows how to read these rungs as information and use it. When you look at a segment of DNA that encodes a specific kind of information, we refer to that as a gene. So genes are pieces of DNA that have information in them. DNA in general is kept in these larger structures called chromosomes. The chromosomes are stored in the nucleus of your cells, and all your cells have the nucleus, all your cells have these chromosomes. And I do want to say that, as we go, feel free to ask questions and we'll try to answer them when it becomes an appropriate time to do so. Especially if it's going too fast and you're like, say that again, or give me some information more about All right, so genes are a piece of DNA that have information in them. That information is instructions for making what we call proteins. Right, so in this, what we're doing is sort of walking through how you make proteins. DNA has the information, and that information, as I said, are those rungs of the ladder. Those are actually chemicals in those rungs, and the cell knows how to read those out as information. And when we talk about that as molecular biologists, we use letters to represent this. So we have the A, C, G's, and T's. Those are the letters of DNA. Those are the chemical, they're representative of the chemicals that make up the information. So your cell has a process of being able to read that DNA and make out of it something called RNA. It's a different kind of nucleic acid, and it basically contains that information that was in the DNA in another form that can be used. So in this case, we've copied that top strand of information into the RNA. If you look, it's exactly the same. So there's the ACC, here's the ACC, here's the GTG. In RNA, we change T's to U, so it's GUG, and on and so on and so forth. Your cell then reads this information and uses this as a code to make proteins. So when the cell sees an ACC together, it says, ah, that is an amino acid called threonine. I'll put that there in that position. The next position is a GUG, that's a valine, I'll add that to the threonine, and so on and so forth. So basically, your cells can take that information in the DNA, make a message out of it, and then encode that message to string together amino acids to make specific proteins. When you think about proteins, you can think of them as machines or signals or structures. So what I mean by that is machines, proteins can do things. Signals, they can signal to other cells or to other parts of a cell to say, do something. Or structures, they can form essentially scaffolds that your cells live with or move with or use to do their structures and functions. Sometimes when we talk about genetics, you'll hear us say a lot of different things that mean the same thing. So when we're talking about DNA, sometimes we'll talk about these as genes, as I mentioned before. RNA is often referred to as transcripts or messages, right? Because it has the information. So if you transcribe this information into a message, you have your messenger RNA or your transcript. We also refer to the DNA as two different things. So the strand of the DNA, the side of the DNA that actually makes the protein, we refer to as the sense strand because it has the information in it. It makes sense. The opposite strand we call the antisense. It does not make the protein. It's basically the opposite code of the strand above it, which does. But you'll hear us talk about sense and antisense, especially when we think about therapeutics. Any basic questions about the nomenclature that we're talking about? Okay, so that's the nomenclature. So how do we get into Angelman syndrome? Well, we have to think about these chromosomes. So this is what's called a karyotype. This is a set of chromosomes from one single cell, all laid out to look at them. This is actually from a genetic test that was done on my son before he was born. So these are his chromosomes, but I'm using them as an example for you. And the chromosome we really care about for Angelman syndrome is this one. This is chromosome 15. And we can recognize it because it has this unique banding pattern. So if you're a person who can read what chromosomes look like, they could look at this and say, oh, that's 15, and I recognize it for these reasons. But you see there's some sort of dark and light structures. All of the different chromosomes have a different banding pattern, this dark and light structure. And people who are cytologists or geneticists can look at this and tell you which chromosome they're looking at. All right, so we care about chromosome 15. And in this presentation, and you'll see it a lot on websites, I'm going to represent 15 like this. So this is one of the 15s. There's two here. I'm looking at one of them. I've sort of illustrated the banding pattern here with some colors. And I've also illustrated the region that we care about the most, which is the region that encodes the information to make UBE3A the protein. All right, so you get one set of your chromosomes. Go back for a second. This is showing you all the chromosomes in one nucleus from my son's cells. And so one of these 15s he got from me, the other one he got from his dad. And that's true for all these chromosomes. So he got one of the ones from me and one of the other ones from dad. He got the X from me, he got the Y from dad. He got one chromosome, one of each of the 23 from me and one of each of the 23 from dad, right? So in me, there was one set in the egg, and in dad, one set in the sperm, and they came together to make the fertilized egg that turned into my child. The thing that's interesting about this whole process is that this egg knows which chromosome came from me and which chromosome came from dad. It has an ability to look at the information on those chromosomes and say, that was dad's information and that was mom's information. And that's going to become more important here in a little bit. But it's really sort of a miraculous process that your cell knows which one is which. So how do we get Angelman syndrome then? So Angelman syndrome is caused by a loss of this function of the protein UBE3A in the brain. So the reason that it's a problem for us is that in the brain, you need UBE3A function for neurons to function appropriately. And Angelman syndrome is caused when you don't have that. So we'll talk about the different ways that that can occur where you don't have UBE3A. And then we'll talk about why this particular form of genetic disorder is really amenable to potentially having therapeutics in the future. All right, so I told you that your cell knows which chromosome is mom's and which is dad's. So in the brain, this becomes really important because on mom's chromosome, which I have here in red, it actually makes a message for UBE3A, which it can turn into protein and use that protein in the cell for the neuron to function correctly. Because of the way neurons are made and the way they function, we turn off UBE3A on dad's chromosome. I can't give you the ifs, ands, or whys about why this happened. We don't really understand necessarily why it happened, but this is the setup. Mom's makes the protein, dad's doesn't. And for most genes in our body, this is probably true. You probably have genes that just don't turn on various chromosomes, but it doesn't matter because the other chromosome can take over and compensate for not having gene expression on both chromosomes. But in the brain, it doesn't. And that's because of this idea of imprinting, where mom's chromosome is recognized differently than dad's in neurons. All right, so what does UBE3A do before we talk about why it's so, what the types of Angelman syndrome are that we have? So UBE3A, when we talked about proteins, they could be machines or signals or structures. UBE3A is a machine. It is part of this complex that works together. So there's three proteins here. There's one called E1, there's E2, and then there's UBE3A, which is also called E3 or E6AP. There's lots of different names for it. We do that in science, and I'm not sure why. I think it's just to be confusing. But UBE3A is this green blob here in this diagram. And its job is to bind to proteins that need to be recycled and put a signal on top of them. So it basically puts this little yellow mark onto proteins that are done, they're finished, they need to be recycled by your cell. This little mark is called ubiquitin. And the ubiquitin, when you put enough of them on a protein, tells the recycling center of your cell, which is called the proteasome, to take that protein and shove it through the proteasome and break it up into individual amino acids. So we can use those amino acids again to make more proteins. We do this to control levels of things that we don't want running amok, right? So I said this is a machine. You know, you want a certain level of machinery to work properly for your cell. If you have too much of it, it can be bad. Sometimes proteins get old and they don't function quite right anymore, so you want to recycle them. It's all the same idea of recycling anything in our lives. Your cells have a recycling program, and UBE3A is really important in this recycling program. And because of the recycling program, you have optimal neuronal functions. This is a neuron. The nucleus would be here at this spot. These are all of the processes of the neurons that send and receive signals. For this neuron to do its job, it needs this recycling program and it needs UBE3A. Now, I want to point out there's lots of different components to this recycling program. This is just one small branch of it, but it seems to be critical for neuronal function, and that's why we have Angelman syndrome. Okay, so how do we get Angelman syndrome? So in our diagram, we've got mom's and dad's chromosomes. And remember, moms can make UBE3A protein, but dads can't when we're talking about neurons in the brain. And so the first way we get Angelman syndrome is if there's a deletion. And in this case, the deletion would remove the information that encodes for the protein from the DNA, and mom can't make the protein anymore. Mom's chromosome can't make the protein anymore. right, so these are actually microdeletions. That might seem funny because we talk about these deletions and they sound like they're huge, but they're actually really small deletions. I've now turned the chromosome on its side to show you some of the things that are in the DNA in this chromosomal region. And what we know is that there are some regions of DNA in this area of chromosome 15 that are just prone to breakage. And this is just part of the way that DNA and molecular biology works. And these are called breakpoints. And if you have a breakage between breakpoint 1 and breakpoint 3, the cell wants to survive, so it will basically repair, but it'll get rid of everything that was in between those two breakpoints and you lose it. That's how you get deletions. And if you lose between breakpoint 1 and breakpoint 3, that's a class 1 deletion, which is what my daughter has. Class 2 deletions are a little bit smaller. They're typically between breakpoint 2 and 3. This is not absolute, depends on where the breakpoints are. There are bigger deletions than this, there are smaller deletions than this. But if the deletion actually removes UBE3A, which is right here, then you're going to have Angelman syndrome if that deletion is on the maternal chromosome. So like I said, these are microdeletions. They are small. So class 1 deletions, which are considered one of the bigger types of deletions, these are 6 megabases. And that sounds horrifically big, but the human genome is 3,200 megabases big. And it has about 20,000 genes in it. So this is actually a very small piece of DNA. Now, I realize that's kind of cold comfort when you're dealing with Angelman syndrome because it's an important piece of DNA. But keep in mind that there's a lot more going on in your kids than just the loss of this gene, and that's a good thing. All right, what are some other ways to get Angelman syndrome? Well, it's the function of UBE3A that's important. It's a machine. So if you have a change in that DNA that makes that information not usable, you can't make a protein that's functional. And in this case, these are called UBE3A mutations. So these are changes within the information that makes UBE3A in the DNA. It's on mom's chromosome. We can't make functional protein, and we don't make functional protein from dad's chromosome. So this also gives us Angelman syndrome. So here's where we get into genetics 201 a little bit. Let me just show you how this works. So with our example from before, here is the information that's going to make this protein underneath it. These single letters are the protein amino acids. And we're going to think about this like a book. So this is like a sentence. UBE3A gene encodes a protein called a ubiquitin ligase. So ubiquitin is that signal it puts on proteins to mark them for degradation. Ligase means added to. So it adds ubiquitin to proteins. So this is the sentence here. And the information is what you just read from the sentence, which is the protein. All right, so what happens with the mutation? Well, that's when you change this code, this 3-letter code in the DNA so that it no longer says what it originally should have said. Sometimes it doesn't matter. So if I sequenced my genes and any of your genes and we compared them, we would see changes between our 2 genes all over the place because everybody's different. And you can use those changes on like ancestry.com or 23andMe to figure out who your cousins are and where you're related and what parts of the world you came from, because these changes don't do anything to proteins, but they are inherited and they can tell you a lot about your genealogy. But when we have a change in the code that doesn't actually change the meaning of the protein, we call this a polymorphism. So sometimes in genetic testing, they'll say, we don't know if this is a mutation or a polymorphism. Polymorphism is just a fancy word to say change that doesn't cause a problem. So in this case, we're going to change ACG to ACC. The cell reads it the same way, so it still puts the same amino acid in that position. So that's a change. It's different, but it doesn't cause any problems. And we could say, okay, well, then we're going to change our sentence a little bit, but not change the meaning of it. So we're going to say called a ubiquitin ligase. We're going to say it's known as a ubiquitin ligase. It's the same information. But some mutations change the meaning, and this impacts the protein. So for example, here, I've changed the CUU to a CCU, and now the cell sees this differently. It's a different piece of information. So instead of putting this L amino acid in this place, it puts P amino acid in this place. And that changes the information and it changes the protein. So now we've changed our sentence. So now we're going to say the UBE3A gene explodes a protein called the ubiquitin ligase. And that doesn't make any sense. You may be saying none of this makes any sense, and I understand that. So if you have questions, feel free to pop them in here. But these changes can be detrimental. So if it makes the information not usable anymore and the protein can't do what it needs to do, you have Angelman syndrome, because now you have what we call a non-functioning protein. Sometimes these changes actually prevent the protein from being made at all. These are called stop mutations or truncation mutations. And that's when you change the code. So here, instead of UAC, we have UAA. We no longer put an amino acid in to make protein. We actually have a signal that says to the cell, stop making protein. So it starts to read this message. It starts to make a protein and it says, oh, I'm supposed to stop, and it quits. And in this case, we've truncated our sentence. We don't even have a full protein. So these are called stop mutations, or sometimes they're called termination mutations or truncation mutations. But in this case, we're not making a protein at all, or even maybe we're making part of a protein, but not enough to function. And then, last, sometimes you can add or delete sequences in the information, and that will change everything too. So sometimes you might be told you have a small deletion just within UBE3A. So you don't have a deletion, you have a mutation within UBE3A, because what happens is if you change this 3-letter code so that it reads different 3 letters than it's supposed to, it'll read that information differently. So in this case, we've added an extra A into the message and it shifts the code. So now instead of reading this as CUU and then ACC, it's reading it as CUU ACC. And now it's shifted everything 1 nucleotide, and now we're getting a completely different protein, but it won't do its job. So in this case, we start out fine, UBE3A gene encodes, but now we've turned it into complete nonsense. So it's making a protein that doesn't have what it needs to be the real protein. The upshot of all of this is that you don't have UBE3A functioning in neurons, and therefore you have Angelman syndrome. All right, the other 2 forms are have to do with the imprinting, the fact that the cell knows which chromosome is mom's and which is dad's. If you have a child that has uniparental disomy or UPD, what happened here is after the egg was fertilized, somehow mom's chromosome got kicked out. It happens. Usually when this happens, it causes spontaneous miscarriages. But in this case, mom's got kicked out and the cell says, wait a minute, I need 2 chromosome 15s. I can't survive without 2. And so it takes dad's and it makes an extra copy of dad's. So now you have something compatible with life, which is miraculous, but the cell sees it as 2 copies of dad's chromosome 15. So now neither one of them are expressing UBE3A. The other thing that can happen is you can have an imprinting defect. And what this means is that that information that the cell uses to know which chromosome is mom's and which chromosome is dad's, that information is incorrect. And now the cell can't figure out which one's mom's or dad's. And so what it does is it shuts them both down and considers mom's to be dad's, even though it's there. In all of these cases, the upshot is we don't have that functional UBE3A. So the neurons don't function appropriately. How do your cells know which one comes from mom and which one comes from dad? Well, it turns out that there are little marks on the DNA. Here I've represented them as circles. This is another chemical called methylation, and there's a different set of methylation marks on dad's chromosome versus mom's chromosome, and the cell can see those differences and say, oh, I see a black circle and a white circle, that's mom. Oh, I see 2 white circles, that's dad. So mom is going to turn on UBE3A, but dad isn't. And I'm going to use those marks to know which one is which. So in most cells of the body, this doesn't matter. In most cells of the body, UBE3A is expressed from both chromosomes. The only place it really matters is neurons, because in neurons, we need to turn on this other message. And this is a long RNA that gets made. It's unusual in that it doesn't actually have information to make proteins. It's just an RNA, but it does have important information. It's important for children, like if you don't have parts of this RNA that gets made, you could have Prader-Willi syndrome, for example. But this RNA is called the long nonsense RNA. It's a long non-coding RNA. We call it the antisense transcript. It's called a lot of different things. But the point is, in neurons, because these marks are different, dad's chromosome turns on this long non-coding RNA. Mom's takes over all of the role of turning on UBE3A. And normally this would be fine. As long as you've got some UBE3A, you're fine. You can think of this long non-coding RNA as a train. It basically runs really hard and fast in this direction. And because of that, the UBE3A gene on dad's side can't really be expressed. The train sort of knocks everything off, and this just sits silent. All right, so what happens in an Angelman neuron? Well, you can probably imagine at this point. So in maternal deletions, for example, we're missing all of that information. We don't even have the UBE3A gene to encode to make protein, but dad still has to make this long non-coding RNA, and this one just doesn't do anything. So that's our problem. We don't have UBE3A in the neurons. Okay. Angelman syndrome and Prader-Willi actually were the first published instances of genomic imprinting potentially being important in disease. So we knew for a while that mom and dad's chromosomes looked different and the cells knew the difference. We didn't know if there was a problem with that. But when we found out that some cases of Prader-Willi and Angelman syndrome were caused by uniparental disomy, then we realized it was important. So if you in your cell, you kick out dad's chromosome and the cell decides to make 2 mom chromosomes for chromosome 15, you have Prader-Willi syndrome. If you do it the other way around, if you lose mom's chromosome and the cell makes 2 of dad's chromosomes, then you have Angelman syndrome. So it matters which chromosome you get. All right, so these are the known mechanisms. Deletion accounts for the vast majority of cases of Angelman syndrome. Mutation is the next largest class. Parental UPD and imprinting defects are a smaller proportion. But these are the general mechanisms. Okay, so when we think about Angelman syndrome, then what are we talking about? Which you all know, but I just want to bring this up because I'll make some points with it in a little bit. So consistent with Angelman syndrome, meaning that if you do not have UBE3A function, you are likely going to have these things. You're going to be predominantly nonverbal. You'll have movement and balance issues. You'll have developmental delay for your skills, and you'll have EEG abnormalities. Frequently, seizures are a part of Angelman syndrome. It's not always true, but most kids, especially deletions, do have seizures. And then there's associated symptoms of Angelman syndrome. Not all individuals with Angelman syndrome have these issues. Some have them worse than others. But we can think of the consistent ones, the frequent ones, and the associated ones. And in general, and I want to be very clear about this, generally, deletions tend to be more severe in terms of how they present with Angelman syndrome than other forms of Angelman syndrome. So deletions present with the most severe effects from Angelman syndrome. Mutations would be next. UPD and ICD are a little less severe, and this is in terms of those consistent symptoms. And then mosaic forms seem to be the least in severity in terms of how they present. So for example, class 1 deletion children typically do have seizures. Mosaic forms of Angelman syndrome less often have seizures. And you're probably saying, what is mosaic? Mosaic is when some cells have the deletion and some don't. And there are some really complicated genetic reasons why that can happen. But in any individual where, say, some of the cells have the mutation in UBE3A and some don't, that's called mosaic because some neurons are fine and some neurons have Angelman syndrome. This is just an in general. This should not be in any way, shape, or form something that you look at and think, well, my kid's doomed because they're a deletion of class 1. I know when Sophie was first diagnosed and people would talk about how UPD was less affected than deletion, it used to just depress me a little bit because I'm like, what does that mean for my kid? The basic take home here is that what causes Angelman syndrome is the loss of UBE3A. Everything comes from the loss of that one protein. But our kids present really differently, right? Everybody's kid is different. You've probably met some other kids with Angelman syndrome that have different issues, or you've heard on Facebook people that are dealing with things that you're not dealing with. You have to remember that there's 20,000 other genes in there that are important in making up who your kid is. I have a friend whose child has Angelman Syndrome. Both of her parents are musicians. That child has perfect pitch. If anybody's instrument in the band is not well tuned, she will leave the room, right? That's not Angelman Syndrome. That's not UBE3A. That's those other 20,000 genes and the fact that she has very musical parents. So we know that our kids are wonderful individuals and they have a lot going for them. So don't get too hung up on the fact that we're missing that one gene. Just remember there's a lot of things your kids can do and nothing is predicted from their genotype of AS. All right, so what should your priorities be? So we're missing UBE3A, what am I supposed to do? Well, focus on their health. That's the most important thing. Make sure you get anything under control that is within your control. That means treat the seizures, treat reflux, anything with GI, make sure your kid's not constipated. Focus on communication, and that means a lot of different things. We have a panel this afternoon to talk about parents' journeys with communication that I hope you can be part of. Help them communicate, and that means sign language or making sure that various vocal sounds mean specific things so that people can learn that, or using communication devices. Anything you can do to make their experience less frustrating for them, go for it. And behaviors, especially when your kids are young, this is a great time to start thinking about how to address behaviors, because what you do when they're young is what you're going to do for the rest of your life. This includes sleep. I think when our kids are really, really young, sleep is very hard for them. And I think it's because their brains are so noisy, those neurons aren't functioning correctly. It makes it really hard to sleep. But as they get older, they start to be able to learn how to sleep better. And there are things that you can do to help your kids sleep. And it's hard. Don't get me wrong, it is really, really hard. But anything you can do to improve their sleep or improve how they behave or improve how they function in their families, this will make your life easier. Which brings me to, what do you need to do? And this is the most important thing I'll probably say to you guys today. As a caregiver, this is hard work. You're not just a parent, you're a caregiver. And that means your health is got to be your first priority. That's mental and physical. Reduce the stress and the guilt as much as you can. Take care of yourself. And that means get some sleep. And if your kid's in that phase where they're not sleeping, figure out hopefully you can figure out some way to still get some sleep, whether it's naps in the middle of the day, or it's whether you trade off with a partner or another caregiver or a nurse or someone hired so that you can get some sleep, because you can't function if you can't sleep. And then your community, find us, right? Find the people who can help you. I can tell you right offhand, like the 4 people that I met very early on in my journey that are still part of my life today that have made such a difference in Sophie's life, I will never be able to repay them. And that's from either just listening or from saying, oh yeah, we had that issue too, try this. And the community's on Facebook, the community is, through the ASF, it's at its conferences. When we can get back in person, it's a great time to meet other people with kids and their siblings and make some friends. Your community is going to be critical. All right, so I'm saying some things about heredity and passing these things on and such like that. Let me just be very clear about this. I am not a medical geneticist, so I am not able to officially tell you about your specific genetic situation and have that be medical advice. I can tell you I am a geneticist, I can tell you what I know. And one of the first things I want you to know is that the genetic changes that cause Angelman syndrome are chance events. They are things that happen because of the way DNA is maintained in cells, the way it's copied, the way that it is replicated, the way that we pass it on to our children. But it's nothing that you did. It's nothing that you experienced or came across or drank or accidentally ate or anything. So for example, let's say that we're talking about my daughter, Sophie. So this would be me and her dad, and here's the egg that made Sophie, and it's deleted. Part of my chromosome is deleted. And I'm a scientist. So when we first got this diagnosis, I panicked a little bit because I'm like, I am around chemicals all the time. I'm not overly concerned about the chemicals I'm around, but maybe that was a bad idea, and I really did get upset about this, and I did a lot of research into it. I realized something that made me feel a lot better very quickly. And that is the fact that this egg that gave rise to my daughter formed when I was still a baby in utero. So all you moms out there, when you were in utero, when you were forming yourself, that's when your eggs formed. So that's when all the DNA got put into the eggs and basically separated. So there was one copy of each chromosome, and a lot of the things that happened before you were even born. So it wasn't something you ate or drank or looked at or, was exposed to. It was, you weren't even born when some of these things happened. I know it's hard to wrap your head around, but the basic thing is the egg that made Sophie formed way back here before I was born. I just happened to carry it on until I was ready to have a child, and that's the one that got chosen out of the billions of eggs that happen to have been made in my body. So in some ways, it's a little bit of a miracle to think about, right? Like, it happened back here, and here's Sophie. So that egg developed when you were still in utero in your own mother. This is true for the deletions. For the mutations, it could be that mutation spontaneously formed when you were making your eggs, but still before you were born. Or it could have spontaneously formed in your dad at some point. Doesn't bother him because he doesn't need UBE3A from his chromosome in his neurons. He got it from his mother. But he passed that mutation on to you where it didn't cause you any problems because it was on your paternal copy. But now it's passed from you as a maternal chromosome into your egg. But again, you didn't do anything. I don't know if that helps you guys or not, but it did help me feel a little bit better to realize that I didn't cause this, right? I could put that guilt away. This just happened. Same thing with UPD, with imprinting. One of the things that life really tries to do is live. It tries to survive. And what your eggs have done in order to make sure that that child could live is pretty miraculous. And, we're missing UBE3A, but again, nothing you did. Someone is asking, could prenatal drug or alcohol exposure cause Angelman syndrome? No. It might have affected other things in development, but Angelman syndrome wasn't caused by that person's prenatal drug or alcohol exposure. It was that egg was already there before they even started to be old enough to drink. So no. Can you pass it on to your children? It depends on what kind of Angelman syndrome it is. Typically, deletions, no. The chance of having another child with Angelman syndrome that has a deletion is about 1 in 15,000. So you'd have to have 14,999 more kids to guarantee that you would have another deletion positive Angelman syndrome child, and even then it wouldn't be a guarantee. And I say most of the time, there are some very rare cases that I'm not covering here of chromosome rearrangements that happen so that you yourself have all the chromosomes, but when you pass them on to your children, they don't get all the pieces. So if you do have a child with Angelman syndrome, it's perfectly legitimate to ask for genetic testing for you and your spouse to make sure that you don't have any chromosomal rearrangements that might have caused this issue that might cause it again. Mutation, it depends on whether or not that mutation is in you or if it's spontaneously formed in your child. If it's spontaneously formed in that egg that was made while you were still in utero, no, you're not going to pass it on most likely. If it's in you, you have a 50-50 chance of passing it on. But this is why you need to speak to a medical geneticist, because they can look at your diagnosis and walk you through the chances of this happening again. Okay. All right, do you have any other questions about the nomenclature that we've talked about or the genetics? All right, so you can bring them back up if you want. I'm happy to stay on afterwards too if you want. Can my kids, when they have kids, can they have AS? Hi, Marilyn. You know what, it depends on what kind of Angelman syndrome they have, right? So my daughter Sophie has that deletion. So now that's in her DNA. And so if she passes that deletion on to her child, yes, her child would have Angelman syndrome. If she passed on the chromosome that didn't have the deletion, they wouldn't have Angelman syndrome. We do have cases where children with Prader-Willi syndrome, girls with Prader-Willi syndrome who have a deletion, pass that deletion on to their kids and their kids have Angelman syndrome. It can happen. Yeah, so, Marilyn, in your case, I would say, yeah, probably, your child with Angelman syndrome, yes, they can have children with AS. Your other children, no, their chances are the same as yours. It's 1 in 15,000. What's the difference between deletion stage 1 and stage 2? So I think you mean the class 1 and the class 2. The difference between class 1 and class 2 is where the break is. Let me just back up for that and I'll show you. So class 1, the breakpoints are breakpoints 3 and 2. It's or I'm sorry, class 2. It's a slightly smaller deletion. Anecdotally, the slightly smaller deletion seems to have less effect in some of those consistent syndromes. So like the class 2 kids seem like their motor skills develop a little faster. Some of them may have less seizures or less frequent seizures. It's not completely clear to me why that is. I don't think any of us know why that is. But you know, in the class 2 case, you're taking out fewer genes. But like I said, all the genes that are here in green are genes that are expressed from both chromosomes. So just because you're missing the green ones doesn't necessarily mean it's a problem, because you're going to express them from the other chromosome. It's just UBE3A and this gene called ATP10A, which we don't think really has anything to do with Angelman syndrome, that it matters if it's maternal or paternal. But the different classes of deletion just refer to where those breakpoints are on the chromosome. And that's really it. Okay. All right, let me. Oh, nope, I didn't want to do that. All right, so let's talk about probably what's really got everybody thinking now, and that is the fact that we're looking at a very new frontier right now. We're in this process of starting to think about therapeutics, and we have industry looking at Angelman syndrome. And I'll just tell you, when my daughter first got diagnosed, she was diagnosed very early at 11 months. And the first person I ended up speaking to, because I'm a scientist, was Art Baudet. And he's a scientist that studies Angelman syndrome. And he told me on that phone call, within 10 years, we're going to be looking at clinical trials. And I was like, what? Because everything I had read made me think that that was it. That, you know, this was the hand my kid was dealt, and that was going to be the end of it, and nothing to think about. But he talked me through the science that night, and I got super excited. And I started with the ASF Scientific Advisory Board that month. So before my child even turned 1, I was already starting to work in this community in terms of trying to think about this. And the goal was always to get industry and pharma to look at Angelman syndrome and say, wow, that looks like a really treatable disease, and maybe we can do something about it. And we're there. All right, so why are we there? Okay, so first you've got to start thinking about what a therapeutic is. And we have therapeutics for Angelman syndrome. And you're probably thinking, no, we don't, but we actually do. So what are they? Well, first of all, we have therapeutics for the epilepsy. We have the anti-epileptic drugs. We have the low glycemic index diet. We have ways to treat epilepsy and Angelman syndrome. We have things to treat the sleep issues. We have the medicines, we have behavioral therapies, we have reflux treatments, we have behavioral treatments, we have motor issue treatments. We have therapeutic interventions for Angelman syndrome. They don't necessarily all do what we want them to do, but they all can improve the quality of life of your individual with Angelman syndrome. And that's what we're really talking about. We're talking about how do we improve their quality of life? How do we make sure that they are living the life they want to lead? And all of these things help. You can't really lead a life if you have epilepsy, right? If you're constantly seizing, you can't contribute. So that's what we currently have. But we think that we can have better ones. And why do we think that? Well, it goes back to the whole way we have Angelman syndrome in the first place. So while Angelman syndrome is all about the fact that we don't have UBE3A in the neurons, what we do have is a perfectly good copy most of the time, sitting there just not being used. And so that says, if we could just figure out how to use that perfectly good copy that's just not being used, we could put UBE3A back in the neurons where it's needed. That's the whole basis for therapeutics, for gene-targeted therapeutics. And so right now there are 2 major strategies that we're using to think about therapeutics. One of them is just to replace UBE3A in the neurons, either turn that copy on that's sitting there so stubbornly off, or put a new copy in there altogether. We could use a virus to go in and put in a new copy of UBE3A and turn it on. Or we could activate that silent allele. And there's lots of ways to do this, including the antisense oligos, the linked nucleic acids, the CRISPRs. There's all kinds of acronyms we can throw here, but the basic idea is that we're going to turn that back on. The other strategy, and it's still a very good strategy because this is the one that has worked for us so far, is to correct the neuronal function in the absence of UBE3A by looking for drugs or compounds that would counteract the fact that we've messed up that recycling system. And there are companies that are working on this right now too. And it may be that we'll need both. We might need to activate the paternal, but also nudge the neurons a little bit to work a little bit better. We may need both things. So when you're thinking about this space and you're like, wow, there's so many companies and they're competing and everybody's doing things, isn't this a bad idea? Never. Competition is never a bad idea, because what it does is it pushes everyone to do better. It pushes everyone to move more rapidly, right? Because you can't wait around when other people are doing it at the same time. So for us as a community, competition, go for it. If you want more information about all these different ways to come up with strategies for therapeutics, I would ask you to go watch the industry updates and the clinical trial videos, especially after having sat through the genetics here, because it'll start to make more sense. What we say to students who come into our graduate programs in science is that everything sounds like a foreign language until you've heard it for the third or fourth time, and then it starts to sink in. So, watch these things over and over again until you feel like you got it. Those trial the industry updates and the trial videos will be posted on the Angelman Syndrome website. I think they're trying for next week, so you can go back and watch everything. All right. Is it reasonable to think that this would even work? And I want to just explain that with the brain is complicated, right? The brain is forming during development, while you're in utero, you've got the brain forming. And we know that the brain basically learns on experience. So as the neurons are forming, as you're experiencing things, the neurons make new connections, they work together. Your brain is complicated, right? How you learn things is complicated. And so when I first got into this, and my daughter was so little, most of the neurobiology people I would talk to would say, you can't cure this because the neurons were already formed and everything was wrong from the get-go. There's nothing you're going to be able to do. Well, we now know that's not true. And here's some ways that we know that that's not true. So first, this was a study that was done with people who were in Art Budet's lab at the time. This included Ed Weaver and Eva Elgersma. And they modified this gene called CAMK2, which is important for learning and memory. And what they found is when they modified this gene and put it into a mouse that had Angelman syndrome, when that mouse was born, it didn't have Angelman syndrome anymore. It still had the mutations that caused Angelman syndrome, but somehow changing this learning and memory gene erased the symptoms of Angelman syndrome in that mouse. This was the study that got all of us really super excited, because this said that that you could modify the phenotype. And part of the reason we get super excited about this is that this gene CAMK2 does not even express until after you're born. So this seems to suggest that the brains are forming okay. What they're not doing is learning, right? So it sounds like, and this sounds horrible, but we've done autopsies on individuals who died who had Angelman syndrome, and their brains seem to form okay. It's not a defect in brain formation. It's a defect in learning. All right. You can restore UBE3A to adult mice who have Angelman syndrome and have the symptoms of Angelman syndrome, and you can actually rescue a lot of those phenotypes. So that says if you put it back, you can restore some of the function that they didn't have. And there is a big study that came out of Eva Elgersma's lab that a lot of people talk about. And in this study, what he did was really clever genetics, but he basically took an Angelman syndrome mouse, and what he did is used a trick to be able to turn on UBE3A at different times. And what he found is if he turned it on before the mice were born, they didn't have any Angelman syndrome symptoms when they were born. If he turned it on right when they were born, he could erase some of the symptoms, but not all of them. If he turned it on later, he could erase some of the symptoms, but not all of them. The important thing to know with this study is that these are mice. And the thing that was rescued at any age was neuroplasticity. So those neurons could function fine at any age when they turn on UBE3A. So for me, my daughter's 15. If I could get her neurons to function correctly and maybe give her a chance at learning things in a more regular fashion, I'm good with that. I think there's a quality of life improvement there. But here's the take home for this. So this, the bottom line is that if you are a mouse and you have Angelman syndrome, we have really good news for you, right? We have all kinds of things we can do to fix the mice that have Angelman syndrome. And this is a shout out to someone who is really important to me, Judah Folkman. But you have to keep in mind that mice aren't humans. They don't behave like humans. They don't learn like humans. The mouse studies tell us that targeted therapeutics are possible, but they don't tell us what it's going to be like in a human. They just don't. So we're not going to know until we know. And the only way we're going to know is when we do clinical trials. So I'm not going to belabor the types of clinical trials there are and things with it. You've heard a lot about this, a lot of the mouse studies and the studies in the lab. This is all the discovery science. There are different phases to clinical trials, and there are lots of good websites, including one that ASF and FAST put together about clinical trials that can talk to you about all the different phases. What I want you to keep in mind are some terms that you're going to hear around clinical trials that are critical, and one of those is outcome measures. So to know if something worked, to know if an intervention worked, you have to have an outcome measure, and that means a test that will tell you what you looked like before you took the drug or before you got into the trial, and what you look like after, and whether or not that change has anything to do with the treatment. And the problem with Angelman syndrome is it's really hard to come up with a good outcome measure. And you might be thinking, well, no, it's not. If my kids started talking in full sentences, that would be great. And you're right. But talking takes a lot of time, right? In a neurotypical kid, it takes 18 months to learn to say stuff. It takes longer to figure out that words mean certain things. We can't expect that our kids are going to get a treatment and start talking in full sentences within a day, right? It's not logical. That's not how we learn. So what are we going to be able to look at in like a couple of months time frame that would show us improvements, that would say this is the way to go, and there'll be those longer term gains over time? We need to measure them right now. Well, one of the difficulties is that we're a very small population. This is a rare disease. There's probably 600,000 people with Angelman syndrome in the world, maybe. And they're not all connected to our community for sure. Not everybody wants to participate in a clinical trial. And so the sample sizes that we have are really small. And so that means we need to have really big changes that we can measure. So some of the trials for drugs that are out there on the market right now, like say for depression, those studies used 4,000 people to see very small changes that were meaningful, but we would never have seen them in a trial with 80 people, which is what we have with Angelman syndrome. So we have to have really good outcome measures that can find those signals and really bring them out so that we know it's working. But here's all the things that we know can go wrong in Angelman syndrome. So which ones are we going to measure? And this is where it comes down to quality of life. What are the things that will make your individual's life better? You know, for my daughter, honest to goodness, if we could help with the movement and the balance disorder, her life would improve immensely. So maybe that's a great outcome measure for her. But she has plenty of friends with the same deletion that don't have her issues. And so a change for my daughter that would be miraculous is no change at all for her friends. So how do you measure that, right? Our kids are incredibly diverse. Well, I'll show you one more thing. I love showing this video. First, I just want to mention that it's great if we can all work on our language and start talking about our individuals as being strongly affected or less affected by Angelman syndrome, as opposed to saying, my kid is low functioning or my kid is higher functioning. This is a judgment. It's not accurate. And it's really about how much does Angelman syndrome affect your child. My child is strongly affected by Angelman syndrome, strongly. She is not low functioning. But this is how she profiles, right? So onset of seizures, 7 months old, started having seizures. She could sit up without support. It took her 3 years to be able to sit upright. And she could only do that for 30 minutes if you sat behind her so she didn't fall over. She only walks with support, and that's still true today. She cannot walk. She was on she tried 6 different seizure drugs. We currently have her on 2, but it took a long time to get those seizures under control. And when we look at some of these tests that we use in clinical trials, she's the lowest possible score. Honestly, I think if you tested a houseplant next to my daughter, the houseplant might do better, right? And so you're probably saying to yourself, well, that sounds like a really low functioning kid. All right, well, let me show you my low function. All right, so this is her. She's heck on wheels on a bike. She can't walk, but she can ride that bike like crazy. And what I want you to see here is that we're in the park, and watch right here. She's going to see this toddler coming in. Your head turn. She likes to run over toddlers with her bike. They make really funny noises. And so I knew that. I'm watching her, but I know what she's doing right now is she's planning. She's trying to figure out how to get away from me and run over that. I'm filming her and I'm watching her. Every time she sees the toddler, she grins. And then you're going to see right here, she's going to go after the toddler, and I'm going to turn off my video. All right. Every measure that we have, she's a low functioning kid. That's not a low functioning child. Right. So what you have to keep in mind in the community is that we have to use these tests. That's what the FDA requires. Some of them are awful and crappy and our kids hate them, but that's what we have. We're going to have to use them. And we're going to have to hope that we get a signal that we can see. So I wanted to mention a couple more things before we're done. And that has to do with trial design in general. A lot of the trials right now, everybody's getting a treatment, and that's great. FDA standards to make something a true treatment usually require a placebo-controlled trial. And with a very basic trial, what you do is you get a whole group of people to sign up for the trial. You use a company that randomizes them into who's going to get treatment and who's not. And you, as the sponsor of the trial, have no idea who's who. And then you do those outcome measures and you see, did the treatment group have a big outcome that the control group didn't have? All right. So why do we need placebo-controlled trials? Because you're probably thinking, I know my kid. I would know if my kid was improved. Of course you would. The problem is that our brains are weird. They're really weird. And they do incredible things because that's what we've evolved to do. What I mean by that is this. Let me give you an example. There was a really intense trial that was done in Parkinson's syndrome. In this trial, what they did is they drilled holes in the backs of the patients' heads, and they used a controlled mechanism to deliver the treatment into their brains. The placebo control group, because there was a placebo control group, had to go through the surgery. They had to have the hole drilled in their heads. They had audio tapes and videotapes that made all the noises so that they felt like that thing was being put into their brains. They didn't get any treatment delivered, right? But they still had to go through that entire big surgery. The thing that was crazy about this is the people that had the sham surgery, who could not walk before the sham surgery, got up and walked after the sham surgery because they thought they got the treatment. If a patient with Parkinson's syndrome who cannot walk, if their brain can say to them, you're going to walk now, and they can do it, you have no ability to prevent yourself from being basically put under a placebo-controlled, I don't know what it would be, like a mirage or something. We can't do it. Our brains won't let us do it. That's the reason we do such strong placebo-controlled trials where the sponsor doesn't know who's on the treatments. The doctors don't know who's on the treatments because we know we can't control our brains. We just can't. So we have to have those trials because we have to be able to say this group didn't improve and this group did, whether we believed it or not. It has nothing to do with you and what you know and your kid. It has to do with the fact that we can't control our brains. This is a joke in pharmaceutical industry, but not really a joke. They get really sad about it. Half the time their placebos perform better than their treatments. This is true for cholesterol medications. It's true for depression medication. It's true for all kinds of medications. It's known. Placebo control is, the placebo effect is strong. It's really strong, right? And you know this. You kiss your toddler's scrapes from their bike wrecks and they feel better. That's a placebo effect, right? You didn't do anything. But it works. And honestly, I'll take a placebo control if it helps me feel better. All right. On the flip side of that is something called a nocebo. And that is when you convince yourself that something is harmful that's not harmful. You'll see this a lot in our community. You'll see people who have decided that, you know, this thing is really harmful to their kid because their kid did X and then had a seizure. Our brains are good at putting things together. We make puzzles. We put them together. But the thing is, unless it's been controlled and you know for sure that those things don't have any correlation to each other, you can't know for sure. In one of the trials in Angelman Syndrome for looking at levodopa, for example, some of the strongest side effects were in the groups that did not get treatment. In the placebo-controlled groups, they reported the most serious side effects from the levodopa, and they didn't get the levodopa. Our brains are weird. That's why we have controlled trials. All right, so what can you do? We need data. We just need to do the trials. So you, as a community, get informed about the trials. You're going to find this on the FDA, has websites about what clinical trials are, what you need to know. There's this wonderful site formed by the ASF and FAST that has phenomenal data on it, as well as data specific for Angelman syndrome. Participate. If we don't participate in the trials and we have those really small numbers, we're never going to see the changes that we need to see in order for something to be approved. You can do things without actually being in a trial. You can save your medical records. You can go to the AS clinics. You can participate in the natural history study. You can put your data into the ladder database. All of these things are hugely helpful. Always, always, always talk to your medical care provider about a trial. Read through all the paperwork. Anything you don't understand, you call the clinical care coordinator for the trial and say, walk me through this. Make sure you understand what you're agreeing to. You are the safeguard for your child. Trials are safe. They have to be approved by a medical ethics board and an institutional review board. They can't be done unless lots and lots of people have looked at it and said it's okay, including the FDA. So in terms of, is it set up to be as safe as possible? Yes. Should you still read everything and make very informed decisions? Yes. And the way to do that is make sure your medical care providers are involved. Let's see. The other things you can do are sign up for the newsletters from the parent support organizations, follow the companies that are looking at Angelman syndrome on social media, and the Facebook communities will also keep you really informed about what's going on. All right, that was the very last thing I'm going to say, and that is, as a community, we have to make sure we don't get in our way. What do I mean by that? So what I mean by that is there are lots of trials from rare diseases where people have gotten together on Facebook in private groups and had discussions about all the great things they were seeing and basically killed the trial. So in Fragile X, there was a trial that was done for a particular drug and the outcome measure was irritability. Did it improve irritability in these children? And that might not sound like a big outcome measure, but it is for them because some of the Fragile X kids cannot go out in the community because of their irritability. They can't be outside of like a very small space. And so to be in the trial, you had to score a certain thing on the intake form to be considered irritable enough to see a difference if you took the drug. Well, a lot of families got together on Facebook and said, if you want to be in the trial, here's how you answer the questionnaire. And so lots of people answered the questionnaire correctly to get into the trial, but their kids weren't irritable. And so they got the drug, but there was no signal, right? Because they didn't have enough irritability to start with at baseline to see an improvement. And so that trial failed. That drug is still not available. The families where they were seeing really good improvements in irritability that were changing their kids' lives are devastated because they can't get the drug now because they basically ruined their own trial. Be careful with this. The companies will say to you, please don't go on Facebook, please don't post on social media, please don't compare what you're doing with other people. It's really hard not to do this, right? You really want to experience this with someone. You want to know if your kid's doing well. You want to be in these private groups. The problem is because our kids are so variable and because it's easy to be suggested to, if you get into a group and someone says, oh my gosh, my kid is holding a ball and he's never held a ball before. Isn't this amazing? This drug must be working. And so you go to your kid and you're like, try to hold the ball. And the kid's like, oh, you've never given me this ball before. I like it. I'm holding it. You're like, my kid's holding a ball too. You don't know if it was because of the drug or because you offered them the ball for the first time. I know that sounds simplistic, but this is going to be critical for us. Just follow the rules. Follow the rules and let's not get in our own way. All right, let me answer a couple of quick questions before we turn it over to Amanda and the diet session. What would I say are the top 3 most do's when supporting children with Angelman syndrome? I had that on the slide. Again, I think it's health, it's communication. Those are the most important things. Behavior. What am I most excited about in the research? I'm so excited about the AS research. I am really excited about the idea of turning on the paternal copy. I would love to see that happen. I would love to see it happen in a way that we don't have to go in and get spinal injections over and over again for that to happen. I mean, we're just at the cusp of this though. We don't know where it's going. but I'm excited for it. I'm excited for all of our individuals and my daughter, and we just keep coming back every year, and we'll keep hearing more and more stuff. Anything else? How does this work? Oh, hi, Iris. How does this work if your child has UPD? Are you talking about the therapeutics? So if you're talking about the therapeutics, the thing with UPD and with imprinting is that we're going to have to be careful, because in that case, you have 2 copies to turn on. And we are aware of some neurological disorders where you have too many copies of UBE3A. These are the 15 duplication syndromes. And there's thoughts that having too much UBE3A could be bad. And you can imagine that if UBE3A is there to help recycle things, you want just the right amount. Too much, you might recycle things too fast. Too little, you're not recycling things well enough. You want the balance. So I think what we're gonna end up doing for UPD kids is we're gonna have to work on things like dosing and control, right? You're probably gonna need a lot less of something to see a nice positive effect in a UPD kid, because you're gonna turn on 2 copies instead of 1. But we're gonna have to be careful with dose. So, and we better get that really right before we try to go in and do something permanent, like, virally insert something. That's my opinion. But I don't think any genotype is going to be left out of this therapeutic wave. There will be ways for everybody to benefit if it's beneficial, but we got to do the trials to figure that out. And Iris, if that wasn't what you were asking, still send me a message. You know how to get me. Yes, there will be a copy of the slides, and the presentation, I think, will be on the website next week. And if you have any questions, any further questions, you can feel free to reach out to me through the Angelman Syndrome Foundation or other ways. I haven't been on Facebook very much recently, but I will try to get myself back on there. But thank you guys for listening to me chatter on. So if you have any other questions. No. All right, thank you guys so much. Oh, thank you guys. I appreciate your kind words. And I would say stick around, because it's amazing what we can do with really good diets for Angelman syndrome, and you're going to hear about that next in this channel. And hopefully I'll see some of you at the next session on communication.